Hello, thank you for joining me. In this short video, I'm going to describe how to count cells using the cell count protocol in Image-Pro. The dataset I'm going to use to demonstrate the protocol is from the Broad Bioimage Benchmark Collection. It has accession number BBBC013. This is a two-channel dataset in a multi-well plate format. This is how the dataset looks in Image-Pro. So you can see we have a plate viewer in which we can see all 96 wells of the plate. And the dataset is two channels, one GFP and one DAPI. And we're going to use the DAPI channel in order to count cells. Let's make a start. So I'm selecting the Count Size tab. This is where I'll find the protocols browser. If I open the protocol browser, you can see that the protocols are arranged in collections. The collection I'm going to use is a cell biology collection, and the protocol is the cell count collection. And I'm going to load the default version of the protocol. Now the first thing I do before I execute any protocol is I click the sync button. This ensures that the protocol is aware of the channels in the dataset you're about to analyze. And I'm going to select the appropriate channel for counting cells, which in this case is DAPI. I'm also going to select the interactive checkbox. And now that I've done these actions, I'm ready to execute my protocol, and I can hit next step. Next, in step 2, we're going to segment out our cells. You can see that this step has defaulted to the smart segmentation option. If I wanted to change to a conventional threshold, I could just click cancel and choose threshold segmentation on the left. But I think smart segmentation is going to be best for this field of cells. So let's select a couple of objects of interest. So I'll start by selecting one of the fainter nuclei in the field, and then select one of the brighter objects. Next, I'm going to select a region of background. This should be all Image-Pro needs in order to successfully segment out my objects. I think it's done a good job there, so let's click OK. Now that we've found all of our cells, we can set a minimum area, so we can exclude small pieces of debris or noise. So here I select the minimum area. Anything smaller than this minimum area will be ignored by the protocol. I could also set a maximum area as well if I wish to, but I don't think I need to in this case. We also have the option of setting clean borders, which means if I choose this option, anything that touches the edge of the image will be ignored. I'm going to leave it and let it count objects that are not whole. I can also, if I wish, set a reference size. This is useful if you have clumps of cells. So if I select a reference object and click OK, and I tell Image-Pro that this reference object contains one single cell, it could now make an attempt, should I have clumps of objects, to come up with a good cell estimate. When I'm happy with my settings, I can hit next step. Here I arrive at the results page. On this page I can configure how we display the data collector results. So I could, for example, choose to auto show the heat map. And I can then configure my heat map to show me the cell count as a mean for each well. I may also wish to show graphs or scatter plots or various other different displays for the data collector, but I'm happy with just showing the heat map. I'm now going to click the read option and hit save and save my protocol. If I check the data collector, we can see what result we got for our first image. We got a cell count of 328. So now that I've configured my protocol in a way that I'm happy with, I can hit finish. This takes me back to the start. I can now deselect the interactive option and hit run selected. This means that I'm going to run the protocol on my active range. In my case, the whole plate is my active range, so I'm going to count the cells for each well of the plate. So let's do that. Let's hit run. At this point, I'm given the option to show the task manager. Using the task manager, I can see that the job has been divided into 6 separate jobs. That's because this is a multi-threaded process, and I have 6 cores on my computer's processor. So by dividing the job into 6, we can speed up the process. When the protocol completes, the heat map is automatically displayed. Here I can see the areas of the plate which had the most cells, which are marked in red, and the wells with the fewest, which are marked in blue. If I check the data collector, I have an entry for each of my wells with a cell count for each well, as well as a snapshot that shows me what Image-Pro has found. I'm also going to display the default report. So I click on the report button. A report for cell counting using my data is generated. Here's the preview of the report. Here we can see a heat map and a graph that shows the cell count for each well, and also the estimated cell count for each well. If I scroll further down the report, I can see my data collector table and also a table of snapshots. This is just a preview of the report. In order to make a full report, I can click Save As and save the report as a PDF. Thanks for joining me for this short video. I hope you found it useful. If you have any questions, please contact Media Cybernetics and we'll be happy to help.